Welcome to lecture MS-2. This is the second lecture of the muscular system. In this, we'll start by explaining what those striations in skeletal muscle are. We'll then get into what sliding filament theory says about how the myofibrils work, talking about the thin and the thick filaments, what's in them, and how they interact. We'll talk about how that relates to the concept of excitation-contraction coupling, which we covered most of in the last lecture. Then we'll cover the contractile cycle, the series of events that explains how myosin produces muscle contraction. And then finally, we'll talk about muscle twitches, their characteristics, how long they last, the relationship between sarcomere length and the tension developed in a twitch, and finally, how twitches summate to produce sustained contraction. Let's talk about the construction of those myofibrils that we find inside the muscle cells. Because these bundles of protein filaments, these myofibrils, they take up most of the volume of the cell, and they're actually what's going to give the cell its striated appearance. So in order to understand that, let's step back a step, back before we were looking at the inside of the cell, and imagine that you're scientists who only have some light microscopes and you're looking at muscle cells from the outside. You've stained them, and what you see, as you see on here, is this striated pattern. So the folks studying this, they decided they wanted to name what they were seeing. So they named the, hold on just a moment. They named the dark section that you see here, right there, they named that the A band. The A stands for anisotropic, which refers to how it scatters light in a light microscope. So that was referring to the dark area. Whereas the lighter areas between the A bands were the I bands, which stands for isotropic. So the structure of the muscle fiber was considered to be this alternation between I bands, which you can see there, and A bands. Like that. So the dark areas are the A bands and the light areas are the I bands. They also saw these faint lines running down the middle of the I bands, which they called the Z lines. So each I band had a Z line running down through the middle of it. Now, that was fine, and what they found, well, we'll get to that in a little bit. So, after people got slightly better microscopes and they could look a little closer, they saw that there were more structures within that. So, let's add in a close-up view using a better microscope. And what you find is that while the A band is still there, this dark region, and the I band is still here, this lighter region, there's a little bit more stuff going on. So, for example, in the middle of the A band, there's a slightly lighter region. It's still in the dark A band, but it's a little bit lighter than the edges. That lighter middle region they called the H band. So what we have now is that there is an H band right in the middle of each A band. And they actually saw that there was sort of a dark line down the middle of each H band, which was in the middle of each A band. So that needs a name too. We'll call that the M line. So we ended up with basically 5 things to talk about: the Z line, the M line, the A band, the I band, and the H band. Those structures form this repeating unit in the muscle that made that striated appearance. And they even they came up with a name for the one unit of this structure that went from one Z line to the next. So this is one sarcomere. One Z line to the next Z line is one sarcomere. So here's another sarcomere. Now, when people first studied this, they didn't know what was making these lines. They just thought, oh, that's neat. And then when people did a little more work, they were able to sort of sample from these bands and try to find out a little bit more about what they're made of. What they found is that if you sampled from the I band, what you found was mostly a protein called actin. There was a lot of actin in the I band. And if you sampled from the A band, what you found is that there was a lot of a protein called myosin. That made people excited because we knew that myosin had some ability to shrink. We know that if you take a piece of meat and heat it, if you cook a hamburger, it shrinks down. Part of that is fluid loss, but part of it is that the myosin protein that makes up a lot of that meat shrinks when it's heated. So people thought, this might explain how muscles contract. Somehow we're triggering myosin's ability to shrink, and that's contracting the muscle, which is a reasonable hypothesis. So like all good hypotheses, it leads to a prediction. The prediction would be that if that's the case, if myosin is getting smaller and myosin is in the A band, then if you look at a relaxed muscle and then look at a contracted muscle, you should see a difference. You should see the A bands getting smaller. That's a sensible thing to think. Let's see what they actually found. So when folks actually did the experiment, what they found was that in the relaxed muscle, you might see bands like this. So I've labeled the A bands in red and the H bands in purple, the I bands in blue, the M and Z lines, and then the length of one sarcomere there in green. So remember, the hypothesis is that when we look at this when it's contracted, the A bands will be smaller because that's the idea that the myosin is shrinking. And what they actually saw was this. Now take a look at this and notice what's the one thing that didn't get any smaller? The A band. So if we look at it, the A bands are just about the same size that they were before. What's different is that the I bands have gotten much smaller. An I band before was this up here, and now it's tiny. The A bands are the same size. The H bands have all but disappeared, and all the Z lines have moved closer together. Now a sarcomere is this long compared to that long. So the question was, so how does this work? The myosin apparently is not changing because the only thing that didn't get smaller was the A band, and that seems to be the myosin. You might look at it and say maybe the I band got smaller, but actin isn't known to be able to do anything like this. So this was very confusing for a long time, until people came up with a theory that we now call sliding filament theory. And here's how it works. The idea of sliding filament theory is that what you see here is actually composed of networks of protein filaments. So I'll sketch this at the same scale that it's shown up in this top one. So what's being drawn on here right now are what we call thin filaments. Each Z line is a backbone for a set of these thin filaments. Over here, I guess we'd have another one. Some thin filaments sticking in here. Move that one down a little bit. between the thin filaments, we will have thick filaments. Like so. And in the sliding filament model of muscle contraction, what's going on in here is that the thick and thin filaments slide over each other during muscle contraction. So this is a relaxed muscle. The idea is that what you see up here with these striations is actually coming from these filaments. The thick filaments are big and block out a lot of the light, so that the area that's the thick filaments is actually the A bands. The idea is that this is the A band. The thin filaments are relatively thin, so they don't block out much light, so that this, the part that's only thin filaments, is the I band. The H band is the slightly lighter region in the middle, which is where the thick filaments and thin filaments don't overlap, so that these darkest areas on the striations are actually the overlapping areas on the filaments. Now let me see if this works here. I don't know if this is going to be functional or not, but we'll give it a shot. Let me save this just in case. So let's see if we can see how these are going to overlap with each other. I'm going to grab this. And see if I can move it. So there you can see those Z lines are actually these actin backbones. The thick filaments are the A bands. The thin filaments aren't really visible. They're too thin to be seen, but they're where the I bands are. And the darkest regions are where the actin and myosin filaments overlap. So that's the idea behind sliding filament theory. And further, the idea is that when this muscle contracts, what's really happening is that the thick and thin filaments are sliding over each other. So let me draw what that will look like. And here you can see it. So just like before, we've gone from the A bands being separated by these broad I bands to being very close together. And in the sliding filament model, the thick filaments haven't changed their size, but they've slid inward toward the Z lines. And then the Z lines have also compressed inward. So the whole structure is just pulling inward on itself, so that the whole thing compresses into a more dense arrangement. That's the sliding filament theory of how muscle contraction works. It's more like going from this, if these are the thin filaments, to this. And if I grab myself a thick filament here, the idea would be that we go from this, where my thick filament is partially overlapped with my thin filaments, to this, where they've slid inward. Now, hopefully that makes a little bit of sense with these diagrams. So there are some questions that remain. One of them is, what exactly is causing this to happen? Why do these work? And second, how? How do the thick and thin filaments slide over each other? What's making them move? Those questions we are about to answer. The first thing I need to do is get into what these thick and thin filaments are. That will let us then talk about what makes them what makes them work together and what makes it happen or not happen, so we can finish up that discussion of excitation-contraction coupling. So in just a bit here, we'll talk about what those filaments are, the thick and thin filaments, and how they're actually put together. Let's talk about what these thick and thin filaments really are. So let's start with the thin filaments. What I've drawn here is meant to be an actin backbone. You can see there the black zigzag, and one thin filament, which here it's meant to look like we're sort of zooming in on it and finding out that it's not just a simple filament, it's actually a twisted double helix, two strands twisted together. And what we see when we get closer is that those are like two strands of beads, and each bead in the strand is one actin protein. So if I looked at an actin protein, what I'd find would be two locations. A site to bind to another actin, so this is the actin binding site, and a location where you can bind to myosin, which we haven't talked about yet. So the actins are stuck to each other, forming that chain, and they have their myosin binding sites sticking out toward the outside of the chain. Now another thing you'll find in here, kind of running down the middle of this, is a thread of a protein called nebulin. Let's just sketch it in here. So that's nebulin. Nebulin acts as sort of a core for this filament, giving it some rigidity, holding it out straight. Now, another important thing on here, running along each of those strands of actin beads is a sort of a ribbon-like protein that sits on the outside. So here it's looping around behind. You see it coming around again, and so on. And we'll do it again on this one. So it's coming out from behind, down the front, going back. So you can see that, hopefully. That is called tropomyosin. And it is held on by this little globular protein. I'll show it in brown. Called troponin. Now, it's important that you be able to envision this in a three-dimensional way. So I'm going to imagine that we take a cross-section right here and then take this and look at it at the end of the filament. So you're looking right down the filament. What you'd see is a central core of nebulin with two actin beads here, there. Those little divots are the myosin binding sites. Now, also on there, you'll find the troponins, and the troponins are holding that tropomyosin out like so it blocks the myosin binding site. Hope everyone can see that a little bit. So if I had a myosin out here, call that a myosin. We're going to talk about what myosin looks like soon, so don't worry about that. Right now, it can't bind. It can't get to the actin. So we don't want that to always be the case. Muscle contraction comes from the binding of myosin to actin. So the way troponin works, let's draw that little troponin in more of a close-up here. So there it's holding the tropomyosin out over the myosin binding site. It has a spot on it where it can bind to calcium. And when calcium binds to it, it's going to move the tropomyosin out of the way. So that means that with calcium, this will go from looking like it does there to this. Our actins aren't any different, but our troponins have shifted to expose the myosin binding site. And now, myosin can bind to actin. So this is with calcium, which binds to the troponin that then moves the tropomyosin. So calcium binds to troponin, and troponin moves tropomyosin, exposing the myosin binding sites on the actin. That's going to happen all the way up and down that filament. So when calcium is there and can bind to the troponin, it's going to move those tropomyosins out of the way, exposing the actin so that myosin can bind to the actin. So suddenly our filament becomes something that myosin can bind to. So that's a thin filament. Now, you may remember when we talked about excitation-contraction coupling that we had action potential in the muscle cell propagated down into the T-tubules, hit the DHP receptor, which pulls open the ryanodine receptor, which lets calcium out from the terminal cistern. And we said calcium that gets into the myofibrils and does something. This is the something. Calcium binds to the troponin and moves the tropomyosin out of the way, so the myosin can bind to the actin. So that is kind of the last part of excitation-contraction coupling. What happens after that is actually the contraction part. That's when we're going to get contraction out of this. So if you're summarizing the elements of excitation-contraction coupling, you could probably say they start with acetylcholine binding to the nicotinic receptors on the muscle cell, causing the graded potential, which triggers a muscle action potential. That propagates down along the sarcolemma into the T-tubules, activates DHP, which pulls open ryanodine, which lets out the calcium from the terminal cistern. Calcium gets into the myofibrils, binds to troponin on the thin filaments, which moves tropomyosin out of the way, which exposes the actin so that myosin can bind to it. So that's excitation-contraction coupling. Now I could tell you that now because now you've seen what the thin filaments look like. So now let's take a look at the thick filaments. So I'm going to make that picture go away, at least that part of it. And now let's bring in what myosin looks like. So a myosin protein has a long tail, a flexible neck, and actually it has two globular heads. It's a little hard to draw that on here. I usually don't bother drawing both of them because the way we're looking at this here, it doesn't actually matter that much. So I'm usually just going to leave off one of those two heads. So anyway, this is one myosin. It has a long tail, a flexible neck, and a little globular head. And that head, we'll draw a close-up of that. Has two parts that we're interested in. It has an actin binding region, here, and an active site where ATP can bind and get broken into ADP plus phosphate. You remember what that does. That's release of energy. It's going to let the myosin do something. Now, these myosins are bundled together into a thick filament. And that thick filament is multiple myosins bound together in such a way that the myosin heads stick out on either end. So this is the idea of a thick filament. It generally contains roughly 200 to 300 individual myosin proteins. Myosin is a large protein, so this thick filament is big. And it's arranged so that the heads are out on the ends. So that we arrange the myosins with their heads out on either end all the way around. So think of it at the end of this thick filament as sort of fuzzy with little myosin heads out here. And so is the other end. Now, let me take this one away and give you an idea of how this would be arranged. So our thick filament would have all these myosins arranged out here where they could bind to the actin. Now, like I said, it's got several hundred, so I'm only drawing in a small fraction of them. The other myosins would be going out the other way, out this way, positioned so they could bind to the other thin filament out on the other side. So we kind of zoomed this out a little. If there's our thin filaments, you have to imagine the actin double helixes there. A thick filament has the little myosin heads out like that. And like that. So all those tails are here in the middle. Then you can imagine another one here. These thick filaments are then sort of held together by some proteins that run up and down the middle. That's actually what makes the M line. Those proteins that hold them together. So our thick filament is mostly made of these myosins, arranged with their heads out on either end, so they're going to be able to bind to actin in two thin filaments on either side of this one sarcomere. Remember the name for this? This is one sarcomere. Now, there is one more protein that's involved here with the thick filaments, and that is the protein titin. Titin is actually the largest protein in the body, as far as we know. One titin protein will run from the center of this thick filament, sort of down the middle here, and attach itself to the Z line, to the backbone for the thin filaments. So that on here would be one titin there, and one titin there. That's T-I-T-I-N, titin. Titin acts almost as a little shock absorber. You'll notice I've drawn it as if it sort of had a spring there. When the muscle contracts and these thick filaments bind, pull, and pull that thin filament in toward the Z line, the titin will get compressed and not let it just jam right into the Z disc. And then when the myosin heads let go, it will help it return back to its resting location. So the titin holds the thick filaments in place in the sarcomere and helps the sarcomere go back to a resting length when contraction is done. Alrighty, so let's sketch up our full understanding here of how something like this might be arranged. So what I've got here is a thin filament comprised of some actin strands like so. We'll sketch in our thick filament here. With the myosin heads sticking out from it. Then over on this other side, we'll sketch in the other side of this arrangement. I'll just skip some of the fanciness here. And then we can sketch in the M-line proteins going up here to the next thick filaments, and the titin anchored here at the center, and to the Z discs. And then on our thin filaments, we should have to draw in the troponin, tropomyosin, and maybe even the nebulin, but that's going to get a little bit difficult to draw at that scale. So that's our sliding filament model. Now, in terms of what it's going to do, so what we hope is going to happen is that when the calcium binds to the tropomyosin and moves it, and the myosin heads can bind to the actin, they will bind and then start pulling on. They will start pulling on this, which will cause it to move in like so. When that happens to both sides, the whole sarcomere is going to get shorter. So the whole thing moved in, the titin compressed, and our muscle contracts. When that happens to all the sarcomeres, all up and down the muscle fiber, the myofibril, and thus all the myofibrils in the muscle fiber, that whole thing pulls inward, which makes the muscle contract. That's the idea behind how sliding filament theory causes muscle contraction. Notice no protein is actually getting shorter, except maybe the titin's getting compressed, but nothing is shrinking. Instead, we have these filaments sliding over each other. Now, all of that depends on the myosin being able to grab and pull on the actin. So what we need to understand now is what we call the contractile cycle, which is how the myosin protein does that. So that's our next goal.